We're going to start another important topic, which is actually HIV. Okay, so let's start. Bismillahirrahmanirrahim. So basically, HIV is a retrovirus, means it is an RNA virus. Okay, it is responsible for the AIDS, which is the last stage of HIV. Okay, so basically the risk is smaller than that of hepatitis B, means the spread of risk is actually less as compared to hepatitis B. It is caused by retrovirus, mainly HIV 1 and 2, which is more, and HIV 2 is actually progressive more slowly and deep in immunosuppression. Mode of transmission is sexual, perinatal, postnatal body fluids, occupational and infected blood products, HIV drug abuser. The most common is basically blood products with we have. In immunology, what happens is basically the virus targets two cells. One is macrophage, the macrophage that actually carries CD4 markers. So virus attack on them. When it attacks on them, then what happened that the envelope of the virus, which is actually glycoprotein 120, binds to the CD4 receptors on the host cells. Okay, when it binds it to it actually, so there are some conformation changes. So they also bind to the another co-receptor, okay, on the cells, on the macrophages or T lymphocytes. Okay, then what happened, they get the entry into the CD4 cells. Okay, then the virus replicate and integrate through the reverse transcriptase integrase, and also they form new viral RNA proteins, and they also kill the T helper cells, due to which actually they reverse the ratio of the helper to suppressor lymphocytes. Okay, so they infect the T cells by either they do apoptosis or immune mediated destruction. Okay, so basically this happens. Then the ratio is actually reverse and antibodies that are produced that are not protective actually now. Okay, so the antibodies are not now protective. Antibodies also indicate that the infection has been now acquired. Okay, now basically all seropositive person must be assumed capable of transmitting it. It means basically that the antibodies which are used for diagnosis actually for diagnosis, which is seropositively, now they are not protecting, but the patient is actually is able to transmit the disease. Okay, there is also means that the person has been now affected. Seropositive means that it can the patient can also transmit and it is also infected. Okay, now basically this is what happened, the immune system T cells depress, and also the depression of the cell mediated immunity. And then what happened to the B lymphocytes? In B lymphocytes, what happened, the polyclonal activation of B cells happened. That results in the large amounts of immunoglobulins. That means actually hyper gamma globulinemia. Okay, it also cause the death cause of death due to immunodeficiency or infection by a great variety of microbes. Now we have some classification of the, you can say, of HIV infection stages. Okay, first of all, the patient is seroconvergent. Seroconvergent, that means basically that the patient is now producing specific antibodies that can be detected into the laboratory tests. And then second means it is asymptomatic. Then the patient actually suffers from persistent generalized lymphadenopathy. Okay, then the patient is constitutional disease have also. This means that it affects the overall health or the condition of the body of the patient. The patient also suffers from neurological diseases. Second, in patients, there is also development of the secondary infections, also cancers and others. Okay, now incubation period, which is actually variable, but it can be in homosexual average 5 years, as long as 10 years can also be.